Thank you very much for the introduction. And also I'd like to thank the organizing committee for putting together this amazing meeting and the opportunity and the invitation to come here and present my postdoctoral research. I'm studying tunicates, and I'm studying specifically germline stem cells in marine tunicates. And first, I want to start with tell you why you are studying tunicates in general. And as you can see here from this illustration, you can see the frog larvae, which is a vertebrate, and the tunicate larvae, the ascidian larvae, which is a tunicate, and we can see how similar they are in terms of morphology. And those similarities that you can see here are actually the ones that led Charles Darwin to hypothesize back in the days that it is very important to study tunicates if you want to understand the evolution and the origin of vertebrates. And indeed, if you look at the evolutionary tree as we know it today, you can see that tunicates are the sister group of vertebrates. So this is the tunicate that I'm studying. This is called Botrytis closeri or Botrytis. It looks like a flower, but this flower is not a single individual. This is a colonial tunicate. This is a colony of several zooids and buds, okay, several individuals. And each petal in this flower, if you look at the illustration that we have here, is actually not one generation, it's actually 3 generations. We have the adult zooid, we have primary bud that connected to the adult zooid, and we have secondary bud that connected to the primary bud. Now, over the course of 1 week, this organism is going through a whole body regeneration cycle in which stem cells are moving from the zooids to the buds and form all the organs within the buds. And then the buds is growing while the zooid is shrinking and just replacing them. And we can see this whole process in the video here, okay? We see how the zooids actually shrink, they are resorbed, and we can see how the new buds are formed, they grow until the zooids disappear. And then the primary bud becomes the adult zooid. The secondary bud is becoming the primary bud. And then we have a new secondary bud that forms. So this process is happening every week. And the regeneration cycle that we see here makes it a really good model to study a lot of things. When we have 2 partners, okay, 2 colonies that sitting next to each other, they can actually fuse blood vessels and form chimeras. Here we can see the blood vessels that are fused, and we can see some white dots. Those are the cells that are being exchanged. And you can see that the whole takeover and regeneration cycle is still happening, while this one is just resorbing. So when we have this organism, now it's a bigger colony. Okay, this is one of the methods of the organism to grow as a colony. But when we take the different tissues within this chimera and we take them to genotypic analysis, we can see that we see representation of genotypes from both partners. However, when we take the gonads within the chimera, we see only one genotype of one of the partners. And that means that actually the germline stem cells in this organism are competing within the chimera to form the gonad. And only the winner genotype will be the one that will pass the gene to the next generation through sexual reproduction. So if we talk about evolution, basically we have a phenomenon here that telling us that there is also natural selection in the cellular level. But what makes germline stem cells winner stem cells, we don't really know. And then my main aim of my study is first to identify the germline stem cells in Botrytis, and after that to understand what controls the germline stem cells fitness, okay? What makes germline stem cells in the winner colonies winner stem cells? So Botrytis are hermaphrodite organisms. Here we can see a photo of the zooid, we can see oocytes, and we can see also testes. The golden areas that you see here are the cell islands. Cell islands, we believe that are also germline stem cell niche, but I can also tell you that are occupied by a lot of phagocytic cells, probably in order to protect the germline stem cells. If you look at the spermatogenesis cycle, okay, this is the cycle that I'm looking for, because when I'm referring to germline stem cells in Botrytis in my project, I'm talking about spermatogonial stem cells, the one that are forming the testes, not the one that are forming the oocytes. So it all starts with spermatogonia, then it's going through a series of mitotic division and 2 meiotic division, 2 primary spermatocytes, secondary spermatocytes, spermatid, when they start to grow the tail, and in the end, what we know as sperm. So the cell that I'm looking for this study is the spermatogonial stem cells. The way to do it is using, the main tool that I'm using is the cell sorter. I will not go too much deep into the technology, but basically cells are going one by one into a very thin nozzle, and based on the morphology, we can plot them on the computer screen. And then we have kind of like a topographic map where we have 3 populations, as you see here, okay, one big here, one here, and one here. The more we go to the right on the x-axis, it means that the cell is larger. The more that we go up on the y-axis, it means that the cell is more complex. And then we can sort the specific cells and just see how they look like and further characterize them. So in the analysis that we did here with those populations, if you look, for example, on the orange population, we can see that those are the spermatid because we can see the round head and also we can see the tail. When we look at the pink populations, we can also see one small sperm cell here. Okay, so based on the morphology, I could tell the germline stem cells that I'm looking for are not parts of this population. However, if I look on the bigger population here that I divided to two subpopulations, one is more granular, one is less granular, I could not identify those cells based on morphology, but I know that the germline stem cells are not here, so I could narrow down a little bit and assume that probably they are found in this big population. Then I was very interested to see how the different populations of the cells in the testes are being developed along the life cycle. So what we did was to take the stage of Botrytis that we could dissect. It's a very tiny animal, so we could dissect the testes separately from the bud and from the zooid. And this is the plot that you already know from the zooid. We can see the three populations, the sperm, spermatid, and what we call Candida germline stem cells. But if we take from the same stage of the colony, but the testes from the bud, we see only this population, okay, the one that corresponding to here. And if I'm taking a look on the different life cycle, okay, the stages that are between those two, we can see how the populations are being developed, okay? So we can see that spermatid population are actually starting to develop first, and then they're becoming more robust, while we can see here a small development of the sperm population, and becoming more robust here, and then more robust here. But the one population that was common to all those stages is the population that I defined in the beginning as the one that will have the germline stem cell in it. This is why I call this population CGSC, Candida germline stem cells. And this is the one that I was focused on. But we wanted to characterize the cells even better. We wanted to do transcriptomic profiling. And for that, we need to do single-cell RNA sequencing. I believe that many of you heard about the 10x techniques. Many labs are doing that. But since I didn't want only to characterize the cells, I want to see, based on the transcriptome, also how I can actually go back to the plot and isolate the specific cells and do the transplantation assays. So use the SmartSeq protocol. In this method, we can actually take lysis plates. Okay, we have plates like 96 well plates with lysis buffer. And using the cell sorter, we are sorting individual cells into each well. We are creating the transcriptome. Then we can see which profile is more interesting. And we can go back to the FACS plot and see where is the location of the cell. Okay, and then we can just go to the cell, to the cell suspension, resort the cells, and move to the functional assay. And when we do that, we have also examination of the transcriptomic profile using the cell clustering. Okay, we can see that the cells that I sorted from the different population, the spermatid, spermatozoa, and GSC, actually divided into 13 different clusters. Each of the clusters is actually representing a common type of cells with a common set of genes that are being expressed. And we have 13 different colors here that representing the different clusters. To confirm that the genes that we have in those lists are actually related to testes, we took some of those genes and we used in situ hybridization. We created probes that are specific to specific genes that should be expressed in the testes. This is, for example, the histone 1 gene. And we can see this is the colony, and we can see the glowing lights here that are actually the testes. And this is a zoom in of the photo with the testes. And also we can use this technique to see the different expression of specific genes on the cells that are in the plot. So if we take, for example, protamine 1 gene, this is a gene that involved in late stages of spermatogenesis, like sperm and spermatid. We can see that it is highly expressed in this area of the plot. So we know the germline stem cell will not be here. We should focus on this area. And the other gene, tubulin 4 beta, which involves in spermatogonia proliferation, you can see that it is highly expressed in the group here. This group is cluster number 2. And then I can go again back to the FACS plot, see where the cluster number 2 cells came from, and keep doing my analysis. And this is exactly what we did. And then we proceed with the transplantation assays after we saw that cluster number 2 was found in the CGSE population. So we took 2 different populations. One was the sperm population. We stained it with green, with DIO. That will be a control because they are not supposed to differentiate any further. They are not supposed to contribute to the gonad. And the CGSC cells were stained with orange. Then we mixed the cell, transplanted it into a recipient colony. And after 8 days, we examined the live samples. And we can see here again the cell islands, the organ that I told you that has a lot of phagocytic activity. We can see that a lot of phagocytic cells actually digested both the orange and green cells. because the immune system of the botrytis is very strong. However, when we look at the gonads on the testes, we could see traces only of the orange cells. You can see one cell here, one cell here. It's not all stained with orange because many of them were stopped by the immune system. But we can tell about those cells that basically we transplanted the cells that we thought that are germline stem cells. It was after 8 days. So the colony actually went through one regeneration cycle. And then in the new gonads, the cells that I transplanted migrated to the new gonads, built the gonads, and also some of them differentiated into sperm. So with that, I will summarize the talk and tell you a little bit about my future plans. That sounds important. So to conclude my talk, so based on index cell sorting, single RNA sequencing, and transplantation assays, looks like we might have in hand the botrytis germline stem cell population. Now, when we know how to isolate them, since we have in the lab colonies that we know that are winner and losers, we can sort the same cells in the winner and loser genotypes and then characterize them based on single cell RNA sequencing and see what factors are the ones that control the competition outcome. In the long term, since again, tunicates are the sister group of vertebrates, so results from this study may be in the future applied in biomedical studies that's related to human fertility. And in the long term, I hope that one day when I open my lab, so I would like to develop pipeline and expertise about isolation of germline stem cells in a lot of species of marine organisms, because I think it will allow us to do gonadial developmental assays that will answer a lot of questions that are basic scientific questions, but also we can apply the science to other applications like aquaculture. And with that, I would like to thank just all my lab members. As you see, we are a pretty big lab, the Weissman lab in Stanford, and also all the collaborators, the funding sources. And thank you for listening.